Hello everyone, today we are going to be talking about glycogen and gluconeogenesis. The learning outcomes for the day are to recognize the 3 steps of glycogen synthesis and degradation. We will understand that glycogen phosphorylase and synthase are inversely regulated, and will conceptualize why allosteric modulators inhibit or activate them. We will describe the structural ramifications of phosphorylation on glycogen phosphorylase. We will recognize new cofactors and reactive intermediates like PLP, biotin, and the oxonium ion. And finally, we will synthesize our understanding of rate-determining steps with substrate cycles and how these new enzymes that reverse pathways must reverse them in mechanistically different ways to remain exergonic. Our Meet a Scientist moment is Lisa Ann Gershwin. She is a marine biologist and has discovered over 200 species of jellyfish. She is in the Australia, New Zealand region doing research there. She is on the autism spectrum and has depression as well. So over the course of this last module or two, we've been thinking about metabolic homeostasis, right? We know that our bodies have evolved to very carefully monitor and modulate a variety of compounds. Most importantly, our bodies maintain the levels of ATP, NADH, and glucose, right? So we've already seen how glycolysis and the citric acid cycle are regulated by levels of ATP or ADP, AMP, and NADH. This is important on its own to provide us the energy we need to thrive. But there are also organs, such as the brain and such as red blood cells, that require a constant supply of glucose and only use glucose as a source of energy. Therefore, our body needs to maintain levels of glucose all day. How is that possible, though, when we don't eat in a while? Right? We have breakfast, we have lunch a few hours later. We're not just constantly eating, so we're not providing ourselves a constant supply of glucose. What if we go to sleep at night? How does our body maintain glucose levels? Or if you fast all day, for example, if you're observing Ramadan, how does your body still survive when you haven't eaten? In comes, thank goodness, our liver. Our liver is the main storage location for glucose in our body, and it's stored as glycogen. And glycogen is a polymer of glucose. So let's talk about what's happening with levels of glucose in our bodies under a variety of circumstances, right? When glucose is plentiful, for example, if we have just eaten a meal and we have plenty of sugar to go around, the excess glucose that we eat is going to be used and moved to storage. So when there's lots of glucose, glycogen synthesis will be activated to turn that glucose into glycogen as storage for later. However, if we haven't eaten in a while, then the availability of glucose is low. That means that first, what's going to happen is that we will start breaking down glycogen from your liver, and we'll convert that glycogen into glucose. You have enough glycogen stored in your liver for about half a day. Once you run out of that source of glucose, then your body switches to gluconeogenesis. And gluconeogenesis is the opposite of glycolysis. And it is the way for your body to synthesize its own glucose from starting materials. You can see some of this on the slide here, folks, right? Oops. You can see here that we can kind of be going from glucose 6-phosphate to pyruvate via glycolysis, or from pyruvate back up to glucose 6-phosphate via gluconeogenesis. Additionally, we can see that glucose can be used and stored as glycogen, or that glycogen could be broken down. Next up, folks, let's talk about how glycogen is broken down and converted to glucose for energy.